All right. Good morning, IMSA.
Morning.
All right, that was pretty good. Welcome to the 2018 IMSAloquium. This is the second time that we have a keynote address, and the first time we're kicking off the event with a keynote. So before we get going, I wanted first of all to welcome everybody. Thanks for getting up early. Thanks to our guests. We have a guest, Dave Ballantyne from NIU. So if you could just wave a little bit. I know you can't see him because it's dark, but he's there. And then our speaker, who I'll introduce in a few moments, and then our research mentors. Before we get started, I wanted to tell you a little bit about how, kind of give you a progress report for the the SIR. And so kind of launch into some boring statistics. Well, actually, I don't think they're boring. We'll just go through these really quickly. This year we have about 145 students involved in SIR, which is pretty good. I think by next year we'll break the 200 mark, so that's even better. This year we have 47 research mentors, and most of them are from off campus. We have research mentors from 3 different states. We have mostly Illinois, as you might imagine. We have one research mentor from Indiana. That's Professor Hederachi. Professor Hederachi, could you just wave to everybody? Yeah, represent. And then one from Wisconsin. And I don't know if our Wisconsin research mentor is here. This year, we're having 8 different workshops. We started the workshop forum last year, so this is an opportunity for you guys to get involved in some of these research projects in a kind of very introductory way to kind of learn about what's going on in the different labs on campus, and also one of the labs off campus. There are 70 presentations, and for the first time this year, we have our on-campus researchers, the adults, giving presentations as well. So there are 7 of those. So that's what's going on in the SIR program as we continue to kind of grow, evolve, change, hopefully improve. All right. Our research mentors come from lots of different institutions, and I apologize to any research mentors if you don't find your institution on this list. I did this from memory. But we have a lot of the usual suspects. This year we have a couple of new comers. We have North Central College, Northwestern Medicine, and Illinois Benedictine University. So we're happy to have everybody here from those organizations. Yeah. All right. One of the things that people don't actually know about SIR is that we spend a lot of money. We probably are one of the best funded programs on campus. And by my estimates, and I think that this is a very low estimate by maybe about 40%, we spend about $1.4 million for this program. Of that, IMSA spends very little. It spends under $400,000 on campus. And those are for supplies and salaries and so forth and equipment. Supplies are kicked in by research mentors both on and off campus. And by and large, what we're getting are essentially the in-kind donations of our research mentors at different institutions. That's actually the biggest expenditure. And if you add it up, that's kind of 80% almost of the amount of money that's spent on this program. So I wanted to say at the outset to any research mentors who are here in the audience, and if you could stand just to make yourself known to the audience, and also in the other locations, just stand up for a moment, make yourself known, and I'd like everybody just to thank them. Let's give them a round of applause. I didn't see one research mentor get up. All right. All right, so today is really about celebrating the work of our students and of our researchers on campus and also of their research mentors off campus. Before we go on, I'd like to thank the people who put today together. On my team, we have Ashley Mann-Lofthouse, who is, you guys all know her. She kind of takes care of you, gets you out of bed, makes sure you get to things, and she runs all the back office stuff. Dr. Wanana Chinwala, who can't be here today, but she's thinking of all of you guys today and wishing she could be here. Dr. Ross Alexander, who just joined our team. These are the people who put today together along, well, I don't think myself, but I'm in there. So, and then we have other people, IMSA team members who were directly related: Kevin Broy, Ralph Flickinger, Dottie Krett, Chris Cornsey, Kathy McDonald, Bill McGrell, and Glenn Yiheng. I hope I said that right. And then we have lots and lots of volunteers. This is truly an IMSA production. So these are our volunteers. I am not going to read all these names. But I wanted to give a heartfelt thank you to everybody who helped put today together and who is continuing to make today a really great day for everybody. So that's basically all I had to say. I'm glad that your kids have had kind of a great start to your research careers, and we're really excited to hear from you. So we'll get to that really soon. All right, today we have a great speech by one of our friends from Loyola University. Our speaker is Professor Nancy Zalesnik-Lee, and she comes to us. She's a professor of hematology and oncology at Loyola Medical Center and the interim director of the Oncology Research Institute at Loyola University Medical Center. And so we talked about this, and I'm not going to read her whole bio, but I'll give you one more important piece. That she graduated from the Ohio State University, where she earned a Bachelor of Science in Microbiology and Immunology, and continued at Duke University, where she did her PhD in immunology, cellular and molecular biology, and her postdoc work was done at the University of North Carolina, Lineberger Cancer Center, where she studied MHC class II gene regulation. So there's more information in the booklet about her, the details of her career, and I think she's going to tell you a lot more about kind of her path. So without any more ado, let's give her a big hand and welcome her to IMSA.
Thank you. It's wonderful to be here. This is going to be a little bit unusual talk for me. Let me see if I can get my slides up. I'm used to giving research seminars, and I was asked to give you a little bit about kind of my career path, if you would, and advice for young budding scientists maybe, or whatever you think your path may be. If I can open this, learning how to be tech savvy on a new computer is... I got it. No? Okay. So it's going to be a little bit of an unusual talk for me, and hopefully you'll bear with my words of wisdom. Because reflecting back on my career path, there are so many things that I actually learned from many different types of mentors along the way. And as I've become now a senior citizen investigator, I actually can appreciate a lot more now, I think, the mentoring that I sometimes fortuitously received along my pathway that really had huge impact on my joy in science and my ability to succeed in science. And I think it's kind of nice to be given the opportunity to young people, some of whom are thinking about pursuing a career, maybe in academia, maybe in who knows what you're going to end up doing. But the things that I think are important maybe for you to pay attention to you as you're starting your journey to whatever you're going to do brilliantly in your career. And so I just wanted to tell you a little bit about my perspective on how to navigate, how I happen to navigate a career in science, and I've absolutely enjoyed my pathway. So I have had many, many different mentors, from family at the very outset to different faculty mentors, all different stages of my career, including now. I have contemporaries of mine who are mentors of mine. And I've actually learned much from my students. So it doesn't matter your title or your position. I think everybody can learn something from their colleagues. So, kind of how did I start? This is kind of an unusual picture, but it represents my parents. My mom was a nurse, and then she ended up teaching nursing. My dad was a scientist at NASA, and he was a physicist and studied stuff I had no idea what he did, but it had to do with fuels. And so I think I had a good kind of support system from a very young age in terms, we talked about science. We didn't talk about physics at home, but we talked about nature, and we just talked about things. And I think it was even a little unusual for my mom to be a working mom during those days, because my friends' moms all stayed at home. And so even though my mom wasn't really, an advocate in the verbal sense for a working woman, she actually, just by doing what she did, provided a strong example for being able to do what she enjoyed and kind of having everything, having a family, having a career. And so I think without really saying many words, and the only picture of a nurse I could find was kind of this unsmiling nurse. My mom smiled a lot more than this picture would reflect. But it just kind of laid the foundation for openness, I guess, to pursuing what you found interesting. And I was convinced at a young age that I was going to be an astronaut. So I grew up in the area where, I was 9 years old when Neil Armstrong walked on the moon, and that was huge for me. And I wanted to be an astronaut. I was convinced that I was going to do that with my life. And my parents didn't dissuade me from that at all. They said, okay, do that, go to school, earn good grades so you can do that. So what I learned from my parents primarily relative to a career path, I think, is their insistence that I work hard and to do something worthwhile with your life. It didn't matter what you did, just make sure it was going to be worthwhile. And so I think with that as a foundation, it kind of gave me a good opportunity. And you students being here, I think you've already had a lot of that foundation. You are good in science, you're smart, and you've had support that allowed you to get here. So this is the beginning, and I hope you appreciate kind of what you have so far and really kind of see that this is already an accomplishment, but it's just the very beginning. You have so much more left that you can do and contribute. So this isn't the end of your careers by any stretch. So as Dr. Kazadi said, I did go to Ohio State University as an undergrad, and after my first year there, I knew I liked science, but I didn't really know what kind of science. I liked biology, and I knew that I wanted to find an opportunity to work in a research lab, because I had never done that. I didn't know anybody really who had worked in a research lab, but I thought that sounded kind of interesting. And I was convinced that I wanted to work with viruses. I don't know where that came from, but I was absolutely convinced that that was what I wanted to do. So I looked up all the people doing research on the campus, found somebody who was working with viruses, and I went and showed up. We didn't have so much email back then, so I just went to that lab and introduced myself and said, hi, I'd like to do research in your lab. I want to do virus research, and I'll volunteer. And he flat out refused me and said, sorry, no. So I could have just kind of given up at that point, been disgusted and said, I'm not going to bother anymore. But I think I took a breath and I walked across the hall, and there was a brand new assistant professor just opening up his lab, and he happened to be an immunologist. So I was like, virology, immunology, I just want to get in a research lab. And so he talked to me. I said, I want to do some research. And he asked me 2 questions. And I think that was my test. he asked me, do you have good grades? I said, of course I had good grades. Yeah, no, I had good grades. And then he also asked me if I wanted to go to medical school. Now, some of you who are thinking you want to go to medical school may not appreciate this, but I really had no interest in going to medical school. And so I was honest, and I said, no, you know, I really don't want to go to medical school. I think I want to be a scientist. I want to do research. And he liked my answers, because he was interested in somebody who he thought really wanted to pursue science. And so he was kind of my first break, if you will. He was willing to take a risk on me, spend his time to teach me things, and mentor me. And along with many things I learned from being in that lab, the first thing I kind of learned was flexibility. I was not doing virology. I was doing immunology now, and I was very happy with that. I didn't know enough initially to be so fixed on doing virology. It was just a whim. I thought it was interesting. I had no real knowledge about it. I may as well learn something else. So actually, I think that was a valuable lesson. Sometimes you're convinced you know something. I wanted to be an astronaut. I wanted to learn about viruses. But sometimes the pathway doesn't have to be exactly what you think it should be. You have to be a little flexible and open. And I think you get many more opportunities that way that end up being really, in retrospect, way better than something you could have linearly planned. So I worked in that lab for actually several years, starting from my sophomore year on. And I learned a lot. I actually got a publication out of it. I met the person who would become my husband later. And I, was a scientist. I learned that doing experiments in the lab, working with other colleagues in the lab was really fun. And I was addicted, if you will, to science. That thrill of actually, doing an experiment and predicting a result, and sometimes you got that result, sometimes you didn't, but it was the process that really got me hooked to thinking, yeah, this is really the right path for me. I don't know exactly what I want to study, but that process really was anchored in my being through this time in a research lab. And so during my undergraduate years, in addition to saying, yeah, this is really fun, I learned a lot about the scientific process, how things work in a lab, what graduate students actually do, kind of how they design their experiments and projects. But I was contributing to science. I was actually this naive undergrad doing my little part of a project. I had a figure in a paper. This was really something to be happy about, proud about. But I also learned that a lot of the personal interactions are also extremely critical in science. I would say everybody who's doing science is smart. That's kind of the baseline. Everybody's smart. They have different strengths and weaknesses, but it's the personal interactions that you really gain the most knowledge from in terms of joy and developing new avenues of research. And so Dr. Mortensen, who gave me a break, I'm still in contact with him. He's retired. He's moved on to enjoying playing golf and things like that. But he actually is extremely proud. Well, that he and now my husband, that he mentored us, and that's actually very important to him. That he had a role in the junior stages of some scientists' careers, and he's very proud of that. And so I think it's our duty to be grateful for those who give you a break, show you kindness by helping to train you, because they don't owe it to you. And they're giving of themselves and of their knowledge to help you move forward. So I think it is important to kind of pause and be grateful for opportunities that you're given, and sometimes try as best you can to pay it forward. That's kind of a catchphrase now, pay it forward, but I think it's true. You were given a break, 20 years from now, hopefully you can do the same for somebody else. I was encouraged to apply to graduate school by Dr. Mortensen, and I applied to several places, and the best place I got in was Duke. He said, you better go to Duke. You got in there. So I ended up going, down south to North Carolina, and I wanted to study cancer. I had personal reasons for wanting to do that. I was interested in chemistry and biochemistry, and I ended up kind of going down a cell and molecular biology, immunology pathway there. My mentor there was Richard Metzker. He was a senior professor, so this was the other end of the spectrum. Dr. Mortensen, brand new assistant professor. Dr. Metzger I actually ended up being his last graduate student. He said after me, he wasn't going to take anyone. No, he was near the end of his career, but he was senior, and he had a very large lab. It was a lab of over 20 people that ranged from undergraduates, graduate students, postdocs, clinical fellows, research assistant professors, the whole gamut, research technicians. So it was a much different experience. With Dr. Mortensen, there were a few people in the lab. The mentor worked in the lab alongside the students. Here, he was much more senior. I met with him every 2 weeks. I was mentored daily by research technicians. I learned stuff from an undergraduate who had been in the lab learning techniques prior to my joining the lab. And it was kind of a more you take your own initiative approach at this stage. There wasn't the mentor meeting me every single day saying, well, you should do this, you should do this. It was up to me to really find out what I should do and how to do it. And the way you did that was by talking to other people. You learned techniques from somebody who had mastered that technique. And it was a much more self-driven type of experience. And so I had meet with my mentor every 2 weeks and say, this is what I've done, these are my results, here's how I interpret them, here's what my plan is for the next 2 weeks. And so it was a very important life lesson in terms of being independent. Because essentially, if you are going to be pursuing science, you have to be self-motivated. It is not you do something because you don't do it, you'll get in trouble, you need to get a good grade. It really is, you're doing it because you're really interested in doing it, and you want to find out the answer, and you want to learn this aspect about the project you're working on. And you have to figure out the best way to do that. And your project is your project. Somebody else isn't necessarily the best person to guide you along that specific path. And so I learned probably the most valuable lesson there was independence. But independence has pros and cons. You take the glory, but you also take the knocks if something doesn't go well. And so lessons are learned. I got publications, just one of them I actually was working on, the original cluster of differentiation groups, the early numbers. Now they're up to CD 200 something, but I worked on CD9 for my dissertation. But valuable lessons from that mentor and that lab were about taking ownership, how to design and interpret your projects, and how to plan ahead. But also to be able to admit when you had made a mistake. Because frankly, it's embarrassing if you make a mistake, and trying to push it off as, oh, it was somebody else's fault, or never works out well. It's better to own up, take responsibility, and figure out how to move past that. I have the same little bit of advice that I give to everybody, all the new students, postdocs entering my lab. Yes, things go wrong. Experiments don't always work. Sometimes you do something really stupid because you just didn't do something correctly. It's better to take ownership of that and say, I think I made a mistake here, rather than trying to cover it up. The experiment doesn't give the data you expected, you can't interpret it, and then you're lost because you're not admitting that, well, I think I put the wrong thing in this tube, or I did the wrong incubation or something, because you waste more time in the end. And it's better just right up front. Yeah, I made a mistake, I won't do it next time. But also that you're not entitled to anything. Nobody really owes you anything. It's on you to be motivated and move forward. If somebody wants to help you, it actually is such a gift, and you should be grateful for any advice or time somebody is willing to spend with you. I also, during that stage, learned to appreciate college basketball. You know, North Carolina is a huge, huge basketball center, and so there's a lot of joy in the lab too. My mentor, who was very kind of stern and unapproachable, boy, basketball season came on, and the Final Four, the ACC tournaments, he had a TV in his big office, and everybody showed up in there. It was so, there's a lot of sides of people, and you can learn a lot, but I think different styles of environments also help you grow, because you may not agree with everything at every stage of your mentoring, but there's some value maybe to take in the back of your mind for when you're on your own, that little nugget actually, I didn't appreciate it at the time, but it actually is important to learn. I then moved across to the other competing basketball team. I moved to UNC, to Lineberger Cancer Center for my postdoctoral fellowship to work with Jenny Ting. She was kind of a mid-level associate professor in immunology there, but she wanted me to join the lab because as a graduate student, I had done a lot of protein purification. I knew my way around purifying proteins, and they needed somebody who had that skill set. And I also wanted to learn more molecular biology. This was in the nascent years of molecular biology. And so I wanted to learn that skill set. And so it was a win-win. They were excited to have me so I could teach my skills. I was excited to be there to learn their skill sets. And I worked on transcription factors, promoter bashing. I learned a lot about MHC class II gene regulation. I was, had a lot of publications through the interactions there. It was a relatively it was a medium-sized lab. It wasn't the size of lab that I had been in as a graduate student, but this lab had like 7, 8 people most of the time. And mostly students and postdocs, and we worked as a team. You had a couple of people that worked on a project. Everybody had a different aspect of the project, but that helped move the project forward more quickly. And so probably the biggest lesson I learned from Dr. Ting was, one of the biggest lessons was, don't be afraid of pursuing a tough question. So we were a medium-sized lab working on something that huge labs were working on around the world, and it was a really competitive area. Gene regulation was just people studying how promoters worked and transcription factors binding to the promoters to drive gene expression. That's what we worked on. And it was really competitive. And some of the labs who were our competitors were these factories. They had 30, 40 postdocs. And I'll never forget Dr. Ting talking to us at a lab meeting and saying, you know, those huge labs, they only have 1 or 2 people working on the same thing that you're trying to work on. So you're not competing against 30 people, you're competing against 1 or 2. So it kind of put a different perspective, is sometimes you want to go after the tough questions because they're important and you want to be doing important work, and you shouldn't necessarily get scared by the competition. You're going to be approaching a problem maybe somewhat differently than somebody else is. You may have a better idea that you can move a project forward better than 3 postdocs who aren't really caring about that project. And so it's good to not back down. If you think something is worth doing, you should do it. But also, you should do it in a smart way. You shouldn't waste your time trying to reinvent the wheel. You should go to the best people who know a technique and ask them to teach you, and pay attention to what they're teaching you. Because that's the fastest way to learn something. Somebody else has troubleshot, they figured out ways to do something better, learn from that, and then appreciate that they've taught you. And another thing I learned from Dr. Ting at that time, she had two little children. So this was a successful, very in the thick of things scientist who's like, you know, it's never going to be a perfect time to have a family. You just make it work. Maybe you won't sleep as much. Maybe, you know, you prioritize. So, you know, for women trying to go into science in particular, you'll have a tenure clock if you're going into academia, but you also have a biologic clock if you want to have a family. And it's something that you can't necessarily decide, I'm going to wait till I'm successful, I'm going to wait till I have enough money, I'm going to wait till I have free time to have a family if you want to, because it'll never be the perfect time. So the converse of that is, if that is important to you, just do it. You go ahead, you live your life, and so maybe you have a few tough years where you don't sleep, or where you have absolutely no time for hobbies. But that's fine. It's decisions and kind of balancing your life. I don't think I slept very much for probably 8 years. I have 2 children. I'm looking very tired. They're 6 years apart, and my second child did not sleep through the night till the age of 5. Well, there's good and bad in that. You're awake, you might as well be doing some thinking about something, but there's joy in kind of having a different aspect of your life. And would I have changed, and would I wish during the time that I had more hours of sleep? Yes, but now in retrospect, I didn't miss that time. I gained so much by just kind of not overthinking it. I had my first son when I was a postdoc. I had my second son when the next stage of my career, which I didn't quite get to yet, when I was a non-tenure track faculty member at University of Chicago. And years later, talking with another faculty member at University of Chicago, in a very candid personal talk we had together, she was more senior than me, and she said her biggest regret was not having children, that she was always waiting for the perfect time and it never happened, and she envied me. She was a much more senior person than me, and she had it all. It was somebody to look up to, but she told me that she envied me, a non-tenure track faculty member, at the time, because I kind of took a pause. It didn't really affect my trajectory career-wise, but it enabled me to have another dimension to my life. And so I actually have talked to other faculty members, postdocs, and graduate students kind of about the same thing, because it's very easy to kind of see go, 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 go. And sometimes it's worthwhile to say, okay, we'll just kind of go a little slower. And you gain sometimes a lot more than you lose by that. So keep your life in perspective, even though you might want to be aggressively pursuing science. So when we were looking for real jobs, after postdoctoral fellowship, we ended up coming back to the Midwest. Our families are in the Midwest. And I was very fortunate at the time because I had my own funding. I had gotten a postdoctoral fellowship that I was actually able to transfer to a lab as long as it was something that was relevant to, it was from the Leukemia and Lymphoma Society. And so that was when I met my next mentor, who I was a non-tenure track faculty member in the lab of Janet Rowley, who was a very well-respected senior professor in the Department of Medicine at U of C. And her lab had just cloned a gene called MLL that would end up now being what I worked on for the rest of my career until now. And they had just cloned this gene, and she was very important for identifying chromosome translocations that were causing different types of leukemia. That's what she was famous for. And they had just cloned this gene at the breakpoint and had no idea how it functioned. And so that's when I entered the lab. I had a background. It looked like it may be a transcription factor, and it ends up it's not really a transcription factor. But with my background, she said, yes, we just cloned this, figure out how it works. And this then became 8 years of my life as a non-tenure track faculty member, working on what really seemed to be a perfect fit for my passion. I had finally found, I think, what I could envision being the science that I was really drawn to. And so this chromosome translocation, indicated here by the yellow, this is spectral karyotyping of human chromosomes, each chromosome a different color stain. You can see there should not be blue and white. It should be just blue. That's a chromosome translocation, a chromosome rearrangement. The MLL gene that they had just cloned was the gene on chromosome 11 that was broken and joined to something else to cause a fusion protein. And on the top is a schematic of the gene that is coded by MLL. It's a 4,000 amino acid protein. It's huge. It has a lot of different functional domains, as we know now, but at the time we did not know that. And as a result of the chromosome translocations, a fusion protein is produced. And so our interest became, how does the wild type MLL protein work? And how, with this translocation and the production of a fusion protein, how does this actually cause leukemia? And can we use this information to try to develop more effective therapeutics? So these are questions that we had when I started, now too many years ago, and I've been working on since then. And the questions are constantly changing, the details of the questions, but the process and the technologies that allow us to address in ever more greater detail and with more knowledge base have continued to grow over the years. This was also the time at which I met Dr. Chinwala, who many of you know here. She was a postdoctoral fellow in the lab, where I was this non-tenure track assistant professor. And what was interesting about that group was that this gene was cloned. We had no idea how it worked, but it was homologous to a Drosophila protein. And it just so happened that Dr. Chinwala, for her dissertation research, worked on the fly ortholog of MLL. And so she found Dr. Rowley's lab, said, "Hi, I'd like to be a postdoc here." Actually, it was a little more complicated than that. Her PhD mentor talked with Janet Rowley, and they said, "She should do a postdoc in your lab." And it was arranged that way. But Dr. Rowley had the openness to realize that somebody working in fruit flies could contribute something important to a human problem. And in me, somebody working on transcription factors and gene regulation could help contribute to the problem. So she brought together people with different backgrounds to try and work together to a common goal. I ended up going heavily along the lines, more recently, of mouse models and really getting into the leukemia aspect. So we developed different mouse models for this type of leukemia that then we could make variants of the MLL fusions to address which specific aspects of the protein are necessary and could be interfered with therapeutically. And that's kind of what I became known for. And so Dr. Rowley was a treasure. She entered science when no women were doing science. She had a backbone, and she had strength of conviction when she identified chromosome translocations and had the gall to suggest that they were what were actually causing the different types of leukemia rather than being side products of cancer gone, messing with the genes. She had to spend 20 years convincing other scientists that this was what was actually going on. And she actually had to stick up for her convictions, supported by data and ever more and more and more data, to finally convince the scientific community that what she was telling them was true, that these translocations were causative of the specific types of leukemia. And so she had the backbone to stick to her guns, supported by her data that she was really sure about. She also had the perspective of being a mom in science. She raised 4 boys while she was working part-time identifying these chromosome translocations. And that perspective that she imparted on us was, you don't have to accomplish everything in your life by the time you're 30. Sometimes you think about things and you can still make progress moving forward. Things have changed a little bit since that time, so I don't know how many people can really work part-time effectively to do science, but kind of the same message is there, though, is if you have balance in your life and you work really hard when you're doing your science, then you should play really hard, too, in the off time. So you have a balanced life. It does give you more perspective and efficiency. You don't waste time, essentially. So another aspect that she taught me was to be humble. This was a woman who ended up getting all the highest honors. Here she is receiving the Presidential Medal of Freedom, the highest honor that a citizen can get in the US, for her work in leukemias. But you talk with her, you see her in a room, she was like your grandma. You know, she was the one saying, you know, I'm not really sure, I'm kind of not familiar with this. Can you explain that to me? When she fully well understood that, she wanted you to realize that you were not explaining something well. But it was her way of not being arrogant, saying, are you crazy? Are you nuts? You're not telling me things correctly. But she would kind of have a humbleness about her that of anybody that I've met, she had the right to be more arrogant. And I think just that aspect, relating from an equal level, even though you may be really super smart, and yes, you've won this National Medal of Freedom, I'm still interacting with you on a one-to-one basis, as you are my equal. Didn't matter if you were the lab technician, an undergrad, a fellow faculty member, you got the same consideration. And that, I think, was one of the major strengths that I learned from Janet Rowley. I've been at Loyola since now 1999, so probably you guys weren't born then. I'm sure you weren't born then, except for the faculty mentors here. And what I have learned then, I moved up the ranks, tenure track faculty member, and these are just pictures of some of my colleagues that I pulled from their web pages. I landed in an environment where I have a lot of colleagues that I talk to on a continuous basis. We have impromptu conversations, we collaborate on projects, and we discuss things in an open atmosphere. And science is really now gotten more and more of a collaborative effort. And if you have good colleagues that you surround yourself with and you take suggestions in an open manner, I think you enjoy it much more, but you also benefit in somebody else has a different perspective because they had different training. So you learn a lot from different people, and these are just some of my colleagues at Loyola. But I also have developed my own expertise, if you will. So I'm known for certain things. And I'm known for mouse modeling of leukemias and structure function studies of the leukemia-causing proteins. So I have something to offer also in terms of a collaborative project. So in pursuing science, it's nice to be part of a group, but you have to have something that you can contribute as well. So it's important to try and at some point develop expertise in an area that people will come to you and say, hey, can you help me do this? Because that actually, in my mind, has been the most fun I've had in the last 15 years, are all the collaborative projects that I've been able to work on. And I'll just highlight, this is a collaborator of mine who we've collaborated for more than 15 years. We have extremely different skill sets. He's a structural person. He does NMR spectroscopy, X-ray crystallography, biophysical measurements of things. These are just figures from some of our collaborative papers together. He does the structure, I do the function. It's wonderful. And we've learned how to communicate with each other over the years to do something that neither of us could do separately. And it's really been very fulfilling to be able to participate in a project that there's no way I do structural stuff. I can look at these kind of plots now and say, ooh, yeah, I kind of see the shifting of that cysteine. But yeah, I trust that my collaborator is good at what he does and will be honest in his analysis of his data. And I've had many collaborators over the years This is probably my favorite collaboration because it's been wonderful. We've also had our non-collaborative collaborators who you go into a project with the idea that it will be a nice collaboration, and it just doesn't work well. So it's one and done. You know, you work together and then, yeah, you know, you don't click. And so kind of finding the people who you can communicate with and move things forward is also a great joy, because it gives you another dimension without you necessarily having to have that expertise. So just a few of our papers that we had together as collaborations. These are just ones with Dr. Bushweller. And so it's kind of my joy in science that we've been able to accomplish a lot. There have been a lot of, this is a big stairs going up. This is my last slide that I have a few pictures coming into it. But these next slides are taken from a family trip that we took, I think 3 years ago to Thailand, when my older son and his girlfriend were teaching English in Thailand for a year. We went to visit them, and you know, kind of the steps up in my mind was kind of the journey that you're going on. It's really sometimes a lot of grunt work and hard work to get up those steps, but when you get to the top and kind of look down, like, wow. You know, in retrospect, you've accomplished something that's worthwhile that you've done. And, you know, you have fun doing it. These are my boys and my husband. And, you know, sometimes you have to be prepared for the fight. Here we were just going ziplining, so it wasn't much of a fight. But, you know, you have to kind of prepare yourself and be strong and go with purpose forward. But kind of this was, you know, we went on a bike ride through the countryside and the rice paddies, part of it. But this reminds me to tell you kind of my realization that I had on this trip, not through my own thoughts, but through my older son. As we were in an airport in Thailand, waiting for a connecting flight, there was a husband and wife couple who were Thai, who had gotten back a few years before from riding their bikes around the world. And this was their dream. And they had done this, and now they're going around giving motivational speeches, and they, you know, wrote books and stuff, you know, go after your dream. And so we stopped to talk to them because they had their bikes there, how they were packed for this trip. So we were kind of looking and talking with them. And the woman came up to us and was asking my husband and myself, kind of, what's your dream? What do you want to do? And I was like, oh, geez, you know, I don't know. I'd kind of like to get more grant funding. You know, I really was feeling stupid because I really couldn't come up with something that I really wanted to accomplish. And I was kind of really being embarrassed there. And my older son, he was like, said to this woman, he's like, no, you don't get it. You just don't get it. She is living her dream. And I looked at my son, who isn't known for being very verbal, you know, in terms of things like that. And I was like, wow, that was profound, because I hadn't really realized that, which sounds stupid. I know I've enjoyed what I've been doing, but I hadn't really had it put in my face that way. That I have really been doing exactly what I would choose to do again in a minute. I really can't think of much I would change other than having more grant funding. Everybody write letters to your congressman to approve a higher NIH budget. But it is kind of nice to look back on the pathway that I've taken, because it was a lot of little things and a lot of opportunities that I was given by people who didn't really have to help me. And now I can say, you know, I really have enjoyed what I do. I still love going into the lab. I don't do so much bench work anymore. The people in my lab would throw me out. I would mess up some things. I still do some of the mouse work, though. But it is fun. So if you have the desire and you think you have really an interest in science, by all means pursue it, because you hear a lot of things now about, you can't make it in science, it's, you'll never be able to accomplish anything, funding levels are horrible, blah. Don't listen, because if it's truly what you're enjoying doing, you shouldn't do anything else, and it should never enter your mind to do anything else. You just take the steps along the way to make it work. And so that's my little spiel about how I think I've followed a pathway that's not so direct necessarily, not the shortest pathway, but it's given me joy, and I can't say that I would do it differently. So thank you for listening, and I'm happy to take any types of questions that you might have, science or otherwise. Thank you.
All right. So, actually, we don't have time for questions.
Oh, I'm sorry.
Because we're right at the end of the talk and we need to start the sessions. Yeah, but Professor Zalesnik-Lee will be here for the whole day. So if you want to catch her and ask her questions, I'm very confident that she'll be happy to answer.